Welcome everyone to the session on design for medical and clinical studies. So my name's Mitra. I'm the chair of this session. The organizer, and as said on the slide, I'm based at the University of Cardiff. I'm a senior lecturer there, but I also have some of my time currently jointly spent at the Office for National Statistics. So we should get straight into the sessions. So we're going to have three speakers. First speaker is Kaspar Rufibach from Roche, followed by Stefanie Biedermann at the Open University, and then last but certainly not least, Thomas Burnett from the University of Bath. So just a brief intro to the first speaker, Kaspar. So Kaspar Rufibach is an expert statistical scientist in Roche's Methods, Collaboration and Outreach Group, and located in Basel. He does methodological research, provides consulting to Roche statisticians and broader project teams, and he also gives biostatistics training for statisticians and non-statisticians. Also mentors students and interacts with external partners in industry, regulatory agencies, and the academic community in various working groups and collaborations. He has co-founded and co-leads the European Special Interest Group, Estimands in Oncology, that has currently 77 members representing 37 companies and several health authorities, and works on various topics around estimands in oncology. Kaspar's research interests are methods to optimize study designs, advanced survival analysis, probability of success, estimates and causal inference, estimation of treatment effects in subgroups, and general non-parametric statistics. So very, very broad and relevant interest to this talk. Before joining Roche, Kaspar received training and worked as a statistician at the universities of Bern, Stanford, and Zurich. So I will pass it over to Kaspar. And just one little thing is I will have to leave at around 2:30 for a little while. So Mona will take over the chairing duties at that point, and I'll be back in case people wonder why there's a little switch, but I will be here for the session. Okay, over to you, Kaspar.
I will talk about answering old questions with new tools, application of the ICH-E9 addendum in oncology, and in the abstract I also promised to talk about principal stratification. I'm not sure I will have time for that, but I hope it will also be very instructive to run you through two kind of pertinent case studies that hopefully illustrate the usefulness of the ICH-E9 addendum. And this material has been presented before, so I want to acknowledge that. I prepared a very comprehensive slide deck that was a half-day course together with Evgeny, a colleague from Novartis. And we drew on the body of work that the Oncology Estimand Working Group has done over the last couple of years. If there's time or interest, maybe in the panel discussion, if you want to learn more, then just ask a question and I can share more information on that. Also, if I guess the slide deck will be shared afterwards, in the backup, there is a presentation of the Oncology Estimand Working Group. I like this quote a lot, and you will see why. So the quote is, "The intellectual illness of clinical drug evaluation that I have discussed here can be cured, and it will be cured when we restore intellectual primacy to the questions we ask, not the methods by which we answer them." And that quote is by Lou Scheiner. When you talk to PK colleagues in companies and also in academia, I think they always in awe when you say the name Lou Scheiner. So I didn't know him, and I have not worked in fields where he was very much involved, but he seems to be like a very prominent and pertinent figure in the field. And he said this quote. And now the question is, when did he say that? Well, it was 1991. So 30 years ago, there was already a realization that maybe we focus a lot on methods, on analytical tools, and sometimes lose sight of the question that we're actually trying to answer. And Lou Scheiner was aware of that 30 years ago, and still it took almost 30 years until, well, the original ICH-E9 addendum was issued, I think, in 1998, I think. So ICH-E9 is kind of the ICH guideline that summarizes the statistical principles to run clinical trials. I guess everybody is aware of that. And 2 years ago, it was amended, ICH-E9, with the Estimands addendum. So almost 30 years after people started to realize that we should focus maybe more on the questions we ask, not just on the methods by which we answer them. And here's another episode. I do a lot of consulting for Roche quantitative scientists, and I get a lot of questions, of course, by email. And there was such a question, and I emailed back and forth with the scientist a couple of times, and just kept asking questions like this very, how do you say, Socratian way, I think, of things, just kind of trying to ask questions to point out maybe the weaknesses or the unclarities. And then the quantitative scientist came back to me and said, this is such a hard exercise. It made me realize I am not sure what exactly I want. And I think this is so typical when we plan clinical trials and plan scientific investigations in general, that we jump on, this is the data we have, and now we want to run some regression models or some other models, and we lose track of the question, what do we actually want to know? So this was an episode that happened to me. Here's an outline of what I'm going to talk about. I will illustrate the usefulness of the ICH9 addendum using a case study in hematology, so a real trial that, in hindsight, we embedded in the estimand addendum framework, and it led to much more clarity. And if we had had the addendum at the time when the trial was planned, I think it would have led to maybe not a different trial, but easier conversations with stakeholders. And the second case study, or this is then more generic, is around treatment switching. And I will conclude with some impact and conclusions around the addendum. And you see here, I have a long backup. So if you're interested in the addendum, why and what does it bring, then I invite you to have a look here. I quickly present the industry working group estimates in oncology. Then one of the methods or the strategies to address an intercurrent event that is introduced in the addendum or introduced to the statistical world in the addendum is principal stratification. And here I give a very short intro into that. What are these estimates and how can they be estimated? Again, interestingly, this is something that has been around in the more causal inference community for 20 odd years. And it took a while until it now made kind of into the statistics world. And then this is related to that. So if you're interested, I don't think I will have time to talk about this, but if you're interested, feel free to have a look. Or reach out to me. I'm more than happy to discuss, refer you to relevant literature, and in case there's interest. Okay, so a case study in hematology. I guess we all know these kind of diagrams. They summarize a randomized clinical trial. And this is the ratified trial, has been published. It's a randomized phase 3 double-blind clinical trial in AML patients. And you see here the two treatments that are compared. So you are randomized, and then you get some induction therapy. If you fail, you leave the trial. If you have a CR, a complete response, so if you respond to the induction treatment, you get some consolidation treatment. After consolidation, if you reach a certain state of the disease, there is the option for stem cell transplant. So SCT is stem cell transplant. And then either after stem cell transplant or, so if you get stem cell transplant, you will not get maintenance, but if you don't get it, you will get some maintenance therapy on top of consolidation. And then you're followed up for the primary endpoint, which is overall survival. Okay, seems pretty straightforward, pretty clear. There is a key secondary endpoint, which is event-free survival. This was the result. So this is overall survival. And you see here that the treatment or the experimental arm did better than the placebo arm. You see here all the numbers, the quantification of the effect. Stem cell transplant can lead to cure. That's why you see this plateauing here of the survival functions. And this is a quote from the paper. Overall survival was significantly longer in the midostaurin group, so that's the experimental treatment, than in placebo, as was EFS, event-free survival, so where you also count relapse and treatment failure as an event, compared to OS, where you just count death as an event. In both the primary analysis and an analysis in which data for patients who underwent transplantation were censored, the benefit was consistent. Now, what is this? This is just a different analysis where it seems there is some concern around what to do with transplantation. So maybe we do different analysis and define a different censoring indicator where transplantation was censored. You can do that. And I think the logic very often is we run a whole bunch of analyses. If they all point in the same direction, we're happy and say results are robust. If some of them point maybe in a different direction, we try to discuss a way why they weren't useful in the first place. So that's how often things, I mean, I'm hopefully exaggerating, but in drug development, that's often how things worked. You had this whole range of sensitivity analyses and hoped that they all point into the same direction, and then results were considered to be robust. But maybe we can have a more structured approach around this. So the question we need to ask is, what question are we actually asking? And let's go back to the protocol. The protocol said, we want to determine if the addition of midostaurin to induction, consolidation, and maintenance therapy improves OS in this patient's population. And the primary analysis was defined as survival regardless, or overall survival regardless of receiving stem cell transplant or maintenance. So in some sense, you consider stem cell transplant as a potential part of the treatment strategy. But then the question is, why would you censor at stem cell transplant? There was a sensitivity analysis where you censor a transplant, but this in the estimand language. So here, kind of SCT is part of an option in the treatment strategy. So you would define your treatment strategy as you add midostaurin to consolidation, to induction, to consolidation. After consolidation, you have the option for SCT, or you go into maintenance. So that's a definition of your treatment strategy. But then if that's your treatment strategy, there's no point in censoring at SCT because it's not answering the question you're interested in. If you censor at transplant, you start to ask a different question. Your treatment effect is then the hypothetical. So you are then, if you censor a transplant, implicitly you start to become interested in a treatment effect in a world where no stem cell transplant was given. Okay, yeah, I mean, that can be a genuine interest, but this has not been said here. It's just basically we did this other analysis and the result was the same. But the question is, or the point is, it implies a whole different question. And that's all okay, but then that should be made explicit. And we should start with the question and not with just doing a different analysis. So these two analyses answer completely different clinical questions. And both, of course, both clinical questions might be relevant, but we should make that transparent, what we are doing here, and to what question a given analysis corresponds. Now, interestingly, we can look into, because this was all in hindsight, so the trial was done and dusted, and approval was achieved in major regions. We can look into what ended up in the drug label in SMPC, so that's in Europe. The label given to midostaurin was in combination with induction and consolidation, and for patients in complete response, followed by single-agent maintenance therapy. Okay, so that's approximately what was on the diagram, but there is no mentioning of stem cell transplant. In the US, it was labeled as in combination with standard induction and consolidation. So here, not only SCT is missing, but also the maintenance is missing. So it seems this protocol objective, somehow you would expect that the protocol objective defines what ends up in the label at the end, because you will not analyze or you will not evaluate something else than in what's in the protocol. So here you run this trial including maintenance, and then in the US you only get the label for induction and consolidation, although whether that by itself has an effect, this is not a question that has been answered by the trial. Okay, so different labels. Stem cell transplant is not part of the label, although it was part of the clinical trial. And the whole point of the estimand addendum is to try to avoid such confusion and bring the discussion of these questions to the design phase and be very clear at the design phase, what questions are we going to answer, what data are we going to collect? Because here, if that would have been the question, we would not need to collect data on stem cell transplant, for example. We would not need to collect data on maintenance, because that question we could answer without reference to maintenance or stem cell transplant. Of course, in this indication, you would still collect that data, but there might be instances where if you're interested in a certain question that doesn't involve a certain kind of data, then you don't need to collect that data. And the data collection should be informed by the question you're asking. So, AML, this is, as we have seen in the diagram, a disease with a treatment strategy that's based on sequence of multiple decision points and treatment modalities. So combination with chemotherapy, you have SCT. But it seemed in RATIFY there was insufficient alignment on what is actually the clinical question that we're trying to answer. And especially when it comes to stem cell transplant. This, as we have seen, I mean, the plateauing, I think, is mainly induced by stem cell transplant. So this is not just an optional thing that some patients get and some not. I think if we want to assess the effect of the treatment, this must be properly dealt with, and this must be an explicit part of the trial, how we want to treat and assess that. So it has a major impact on the benefit risk of midostaurin. But how to treat it has not been outlined in the trial objective. For maintenance, things are a bit different. It has been mentioned in the trial objective. It ended up inconsistently being included in the two labels. Now, of course, it's easy. Now we have the estimand addendum. It gives us a tool how to design trials in a better way. So we can go and say, okay, if we designed RATIFY today, how would we do it? Well, we would first say the clinical trial objective is the addition of midostaurin to induction, consolidation, and maintenance with the option to receive stem cell transplant in CR, whether that improves OS in AML patients. So this is not so different from before, but now the estimand addendum will make this clinical trial objective more specific by defining 5 attributes. The first attribute is a clear definition of the treatment strategy. And now I think this adds value here because that was not so clear before. So here you specify what you get in induction plus midostaurin, that's the experimental drug. And then what you get in consolidation in patients with a CR after induction. Then you get midostaurin maintenance, and after consolidation, you have the option to receive SCT. And the same in control, where you replace the experimental drug by placebo. So that's the definition of the treatment strategy. So here, SCT becomes an explicit part of the treatment strategy. And if you then have a successful trial, that must end up in the label, because that is how you define your treatment strategy. You cannot just ignore that. Of course, health authorities can do whatever they want, but the idea would be that the label then matches what you define here as your estimate. The population is pretty straightforward. The variable is overall survival, the endpoint. So kind of the endpoint becomes a part of your estimate. And then intercurrent events. So what are intercurrent events? Is there a question? No, because I cannot see you guys. I just see my slides. So if you would like to ask a question, then please do so. Just unmute yourself and ask.
So we'll be monitoring the chat. So if there are questions, we can let you know, Kaspar, as well.
Okay. Oh, that's good. Yeah. So I'm more than happy to answer questions. Apologies, I forgot to mention that initially. So please feel free to ask. So what we call the endpoint becomes one estimand attribute. That's the variable here. That's overall survival. And then intercurrent events. These are events that happen between randomization and the evaluation of your endpoint, so OS, or between randomization and death of a patient. Clinical events that may alter or preclude the observation of overall survival. So for example, if you had not included stem cell transplant in your treatment strategy, then stem cell transplant would be an intercurrent event because of course, stem cell transplant alters the interpretation of overall survival. Okay, but now we have absorbed it into the treatment strategy. So in fact, in this scenario here, there is no intercurrent event left for overall survival. We have integrated all these decision points or these clinical events that can happen into the treatment strategy attribute here. And then the summary measure would be the hazard ratio. This is something we can discuss when we see how the things plateaued. So this is of course not proportional hazards, but the hazard ratio remains an approximate useful summary measure in any case. So this is how we would most likely define the estimand today in Ratify. In hindsight, given of what we have extrapolated was of interest to the investigators at the time. So to wrap things up, this is a very introductory example. If you have such complex multi-phase strategies, you can have on top of it non-proportional hazards. We have seen that when you look at the estimates of the survival functions. You can have cure. So these are additional features you need to think about that these might affect the summary measure. So at some point you might end up saying, well, I'm not sure the hazard ratio is the appropriate measure to summarize the effect. Maybe I should take something else. But all this has something in common. So the fact that we have stem cell transplant, that we have these multi-phase strategies, that we have potentially non-proportional hazards, that we have cure, they have all one thing in common. They can all be anticipated. You knew about every single of these aspects when you designed the trial. So we should be clear about what we are going to do. So this should enable us to clearly formulate the clinical trial objective. And that is key. And then once you have this clinical trial objective, this is more kind of a verbal statement of that kind of introduces the protocol most typically. And from that, you make that then more specific with the estimand definition. And the estimand definition in the sense of the ICH E9 addendum are these 5 attributes that I listed for Ratify. Okay, and what this should achieve is that many of the discussions that we have, so it should of course achieve many things, but one key achievement is many of the discussions we have when we see the results of a trial, how to interpret that, and then which clinical event to censor how, and should this be an event, that should all happen at the design stage and not at the stage of the analysis. Because if you have this at the design stage, for us in drug development, that saves a lot of time during the filing, and also it hopefully avoids disagreements with a given health authority because they thought you're going to do A, but you thought you're going to do B, but because you were not so clear about it, A was not equal to B, and then you have this discussion once the data is in. And then if what the authority requires is maybe something for which you haven't collected the data because you have planned for something different, that can bring you in serious troubles. So that's another big achievement if you have these discussions up front. If your clinical trial objective and your estimates clear, it should also be clear which data you need to collect and which data maybe you don't need to collect. Okay, so that's a key implication as well. And then also when you have an estimand defined, the analytical method by which you estimate that estimand should also be defined up front and should be clear. And then you should also be clear what data you need in order for you to be able to compute that estimator. Okay, so we are seeing big steps in that direction, and the big advantage now having this addendum leading to better trials, easier communication, more clarity on data collection. This is in the pharmaceutical industry. I cannot speak so much about the academic environment, but my sense is that it has not kind of permeated the academic environment to an extent it has pharmaceutical industry, but I'm pretty sure it will over time, at least when also, for example, journals will start to require a clear formulation of an estimand. So this is something to watch out also if you work in an academic environment in clinical trials. If you're interested to learn more, I invite you to go to the Oncology Estimand webpage, so oncoestimand.org. There all publications are listed. So this is just one publication that discusses this ratify example and 2 other case studies in a similar context, so in hematology. And it gives you templates for protocol and SAP, statistical analysis plan, formulations of the primary estimand and sensitivity analysis. So if you're interested or just want to learn more about this, I invite you to have a look. Maybe I pause here for a minute. Any questions to this case study, to this example? Okay, maybe we can discuss questions in the panel discussion. So then I will briefly share a second case study, treatment switching. And I start off with good old days, Herceptin. So this is a drug to treat HER2-positive early breast cancer patients, and was assessed in a trial, large trial, HERA, where the primary therapy was surgery, chemotherapy, or radiotherapy, as indicated in early breast cancer. And then what the interest was in a comparison that after completion of this primary therapy, giving Herceptin versus just observing the patient, whether that has a benefit. So randomized phase 3 clinical trial, disease-free survival was the primary endpoint. The trial was stopped early for efficacy at the planned interim analysis in favor of Herceptin or trastuzumab. Now, at the time, these were the options the patients had. So there was no treatment after this primary therapy, and there was no alternative treatment to Herceptin. So, of course, when the trial read out positively at an interim analysis, all patients in the control arm were offered the option to cross over to trastuzumab. And eventually, over the long run, about half of patients actually did so. That, of course, has an impact on the effect we estimate. And here you see the impact. So this is DFS, the primary endpoint. This was when the trial was stopped. So median follow-up was 1 year here. This was when the trial was stopped, and before stopping the trial, nobody had crossed over because it was not clear yet whether Herceptin is doing something. The hazard ratio was 0.54. Okay, these were the number of events. But then over time, control patients started to cross over. Okay, so here 4%. This is the percentage of control patients that had crossed over. And of course, now you get kind of a mixture in the control arm. You will have patients who got the primary therapy and then have just been observed until their DFS event. But you will also have patients who get their primary therapy and then started or crossed over to trastuzumab. So they basically got the same therapy as the treatment arm. So your estimated benefit will be attenuated. And that's what you see here over time. So the hazard ratio is moving up from 0.54 to 0.76, but here it remains quite persistent. Okay, and you see, I mean, this is a very long-term story. So over 10 years, we kept collecting the data, and here you see the DFS hazard ratio. And the same for overall survival. So this is early breast cancer. So luckily, not too many deaths for a long time. Here you see that you only had very few deaths at the time of the primary analysis. And then later on, despite crossover, even a better effect. But then here again, you have attenuation, and it zoomed in on about 0.74 now with quite a few effects, but with half of the patients crossing over. Okay. And the question is, okay, what effect are we ultimately interested in, and what can we do about this crossover? And here, I think what we should recognize is that we were able to establish the overall survival effect despite 50% of patients crossing over in the control arm. So basically what we did is we just had like 1/4 of patients who got the primary therapy plus the observation as specified in the protocol, because half of the control patients crossed over at some point. And still we got an overall survival hazard ratio with a confidence interval that you can say, okay, it doesn't cover 0. So we established the overall survival effect still. So why was that? Well, Herceptin was a new drug class at the time, an antibody. You had a large treatment effect, even on overall survival. You could basically afford all this crossover, okay? Because, I mean, 25% reduction in the risk of death is huge by itself, even without crossover, and that could even be established with half of the control patients crossing over. These control arm patients, they didn't have another option. Once the trial had been unblinded and the Herceptin had been approved, crossover represents standard of care. It is basically what you immediately become interested in, even for patients in the control arm. Okay, and that applied globally, because globally there was no alternative therapy for the patients in the control arm. So this treatment policy estimand, this is what we are looking at here. You have this intercurrent event in the control arm that patients cross over. So remember, we defined an intercurrent event and a clinical event that alters or precludes the observation of the endpoint. Now, of course, if you switch to Herceptin out of the control arm, this is a clinical event that alters the interpretation of overall of your time to death. So this is an intercurrent event. And when we compute overall survival by just looking at until the patients died, not accounting for the fact that half of the control patients have crossed over, then we say in the addendum language, we use a treatment policy estimand. Okay, and because of these reasons, this estimand is interpretable here. But the oncology landscape has dramatically changed since the good old Herceptin days. This is looking at the clinical trials with anti-PD-1, PD-L1 agents. And you see, I mean, this is just exploding. And just today in New England Journal of Medicine, a comment by Pasteur and another colleague, I think from FDA, appeared, where they basically say it's absolutely overwhelming how many such agents are today developed, and the whole development is uncoordinated. So if you're interested, you can have a look at that. But just to say here, I mean, you have all these agents, and these are all the indications where they've been developed in. The same is true for CAR-T. Many different CAR-T approaches being developed. Now, what implications does that have? Well, it's great for patients. You get durable responses, and you have many ongoing clinical trials where patients can get on. But what does it mean for running clinical trials? Well,
sorry, Kaspar, to interrupt, but if we can wrap up, would be great. Thank you very much. A couple of minutes. Thank you.
Okay, maybe another 5 minutes or so. That's fine.
Okay, thank you.
If you have this situation, so you randomize 2 treatment strategies, you have end of therapy, and then patients get some other therapies later, and you follow them up till death. The typical OS definition is just time from randomization to death, regardless of the journey of the patient. You use a treatment policy for every intercurrent event. So if patients cross over from one therapy to the other, get some new therapy, etc. If this reflects clinical practice, what's happening here after end of therapy, then this is approximately interpretable, because that's what patients would get anyway in the real world. Okay. The same is true if, for example, you compare here drug A versus standard of care in first line, and drug A is already approved in second line. So a patient in standard of care after end of therapy, they will get drug A. But again, if this is established standard of care, just looking at overall survival remains interpretable, because it reflects clinical practice. And that's what we ultimately are interested in. Although you're not estimating the true biological drug effect of this initial randomization anymore, but you estimate an effect that you can expect in clinical practice. It starts to change in this landscape where patients on the standard of care, when they fail the treatment or after end of treatment, move on to one of these myriads of other clinical trials with, for example, the very similar investigational drugs with the same mode of action. Because these drugs are not approved yet. If you then just look at overall survival, you're not assessing anymore clinical practice, okay? So the treatment policy, so the subsequent therapy after end of treatment does not reflect clinical practice. This is what we see in immuno-oncology, CAR-T, everywhere. The question then is the treatment policy estimate still relevant? Or would we not rather be interested in something like the benefit on overall survival without crossover, in a world where this crossover or this change to a not yet approved drug would not happen? And this is what we call a hypothetical estimate. That might be of more interest, even to decision makers, even to health authorities, because what do I gain from learning about an effect that's not clinical practice? Okay, maybe these drugs will never be approved, and then they will not get these drugs. So it's not clear how to interpret the effect I get. You can estimate this hypothetical estimate with certain statistical methods, but I wrap things up now. This is another feature I will skip. I think what we in pharmaceutical industry are concerned about, if these subsequent therapies do not reflect clinical practice, drugs are not perceived, are perceived as not improving survival. And here are just a few headlines. While they are maybe not kind of properly assessing the effect that we can expect in clinical practice. So this makes a case for a hypothetical estimate that can be meaningful in these scenarios, and that's what we wrap up here. So if you're interested, feel free to have a look. This is also something we, out of this cross-industry working group, we put together estimates for overall survival in clinical trials with treatment switching in oncology, and there we kind of discuss all these scenarios and also how you can estimate such estimates. This is just kind of a recollection of methods. These methods have been developed earlier, so this is not something we developed. And let me just go to my last slide, impact and conclusions. So the estimate dictates the data that needs to be collected. It requires a multidisciplinary involvement from earliest stages of clinical trials. So statisticians in clinical trials should not let that push to them that basically you are the statistician, you write down the estimate. This needs discussion with everyone involved. It impacts the design of the ECRF or other data collection tools, of course, also the monitoring strategy. Depending on the strategies you use for intercurrent events, it might need increased effort in recording reasons for why patients leave a trial, why patients stop a treatment. You might need to reflect the estimate assumptions in sample size computation. And I think with this, I close. So I hope these two examples made it a bit clear that the estimate addendum is useful. It brings clarity to questions, and that it shortens many discussions after readout and brings them to the design stage where they are easier to have and alignment can happen, which ultimately makes life easier for everybody involved. And I like this quote very much by Don Rubin: design trumps analysis, so don't jump on analysis methods. First, think very carefully about the design of your experiment. And with that, I close, and I look forward to the rest of the meeting.
Thank you very much, Kaspar, for the very interesting talk. We are short of time regarding questions now, but we might take the questions for the panel later on. So please, if you have any burning questions to Kaspar, drop them in the meeting chat, and then we can take them at a later stage. So moving on now, I would like to introduce our second speaker, Professor Stefanie Biedermann, who is a professor of statistics in the School of Mathematics and Statistics at the Open University. Her current research interests center on the area of optimal design of experiments, and in particular the development of methodology to improve statistical designs and analyses when data are incomplete. and she will be talking about this topic today with us. She currently holds an ESPRC grant on designing experiments when data may be missing, not at random. And Stefanie had her PhD from Germany and has been an associate professor at the University of Southampton before moving on to the Open University. So it gives me pleasure to introduce Stefanie, and she will be talking to us today about the optimal designs for multi-arm trials with dropouts. Thank you very much, Stefanie, for coming forward, and over to you.
Thank you very much for inviting me. It's an honor to speak here. Let me share my screen. So today I would like to present some joint work with Kim Lee and Robin Mitra. And I'll give a brief introduction to the problem. I'll then talk a bit about models for longitudinal trials, what kind of design of experiments we are talking about, and then I'll show you some results. First a toy example, and then an Alzheimer's trial, which is a bit of a work in progress, really. Okay, so let's start with what is a longitudinal trial, or what does it typically look like? Okay, here at the start we have patients. Usually they have something measured at baseline, then several interim responses, and then a response at the end. So that's the ideal case for the trial, but then in reality what we usually will experience is that at the end of the trial we will not get responses from all patients, and even at some of the intermediate time points we might have dropout already. So today I would like to talk about longitudinal, potentially multi-arm studies with potential monotone missingness or dropout, which means that if a patient missed one appointment, we assume they will miss the subsequent appointments as well. Now, the usual mitigation strategy for dropout would be to increase the overall sample size, which is of course a good idea to do. But we are thinking, can we do something a bit more elaborate to make the experiment more efficient? And the question was, can we use design of experiments techniques to improve the accuracy of the statistical conclusions here? Okay, now let's consider what would be part of the design of the experiment. Now let's have a look at the situation here. You can see some of the patients have turned green, which is just to indicate that they actually have been given a different treatment. So now we've got these two different treatment groups, and we notice that there seems to be more dropout in the green patients than in the other patients. So there might be different dropout for different treatments. And so the question is, should we maybe, instead of just having equal allocation in all groups, should we maybe have some more patients in the arms where we can expect more dropout, which seems intuitive? And we know the proportion of patients or randomization ratio is part of the study design. So we could incorporate differential dropout into our optimal design framework. Okay, another part of the design would be the time points where we take the observations. And here what is also possible in optimal design would be to consider different time points for different groups. But in medical applications, in clinical trials, this might be not very useful, because then you would have issues with blinding. You can't have a double-blind trial and then still have patients at different time points. But generally, that is also an option. But now about changing the time points in general. Okay, is that a good idea? We might consider bringing the time points slightly forward. So we still have the same dropout at the end, but maybe some patients will not have dropped out by the time we are taking some of the other measurements. So time points where we take measurements are part of the study design. Bringing some of them forward may give us more observed values of the response. But then we also have to incorporate that into our criterion that measuring a patient too early might also not give much information, neither from a statistical nor potentially from a clinical point of view. In particular, if it's a more slow-acting treatment, we wouldn't want to measure too early, but then measuring too late might not yield a response at all because the patient may have dropped out. So here we also need to strike a balance, and we would try to do this through design. Okay, finally, what is also part of the design would be how many observations can I take per patient. So here I've added in another time point where responses can be observed, which may be a good idea in order to get more observations per patient, in particular when some of the patients are dropping out later. But then there's also cost involved of having patients coming back to the clinic, for example. Therefore, we might have fewer patients overall in the trial. Or if we want more patients, we might have fewer time points in the middle. Okay, so the number of time points at which we observe responses would also be part of the design. And for a fixed budget, we can then either have more time points and fewer patients or vice versa. And an optimal design could also try to strike the right balance within clinically reasonable bounds. So it would have to be spoken about with clinicians, how many time points are maybe the minimum or the maximum they would think reasonable. Okay, now what kind of models are we talking about here? In longitudinal trials, we would consider mixed models. And today I'm talking about linear mixed models. So the responses for patient I are in a vector yi. Then we've got this fixed term regression part where xi is the design matrix for patient I, and beta is the fixed or population parameter that we want to estimate. And then we've got a design for the random part, we've got a design matrix zi and the vector of random effects bi plus some vector epsilon I, which is the random error. And we are assuming that the BIs are IID and normally distributed to mean the zero vector and some covariance matrix D. And the epsilon i's are also a vector now, since y is a vector, normally distributed with zero mean, and some covariance matrix, which may be just the identity matrix, but it could also be some more complicated structure to incorporate serial correlation in the responses from the same patient. And the usual assumptions are that the BIs and the epsilon i's are also independent of each other. Usually the main interest is in estimating beta, because that's the vector of population parameters. Or it could be a subset or a function of beta, for example, some contrast that is a treatment effect. And we also have an unbiased estimator for beta for maximum likelihood, and we have a closed form for the covariance matrix as the inverse of the Fisher information matrix. We can write the Fisher information as the sum of individual information matrices for all patients, where VI is the covariance matrix of the vector yi for the ith patient. Or we can also write this in terms of groups. If we have C groups, at nk is the number of patients in group K. Okay, so that is for the case when we expect to observe everything. But then, what happens if we have dropout? We assume that the probability of dropout will depend on the time point. So the later in the trial, the higher the probability of dropout. And it will also depend on the treatment group. So if we define P-ops as the probability of observing a response for a patient from group K at the jth time point, then we can also work out the expected number of patients in group K who have exactly J responses as MKJ, which is the the initial number of patients in the group times the probability of observing at point k minus probability of observing at point j plus 1. And this will help us to find the information matrix with dropout. If we just define a design matrix xkj and a covariance matrix vkj for the incomplete vectors of responses for patients who have exactly j responses, then we have a closed form expression for the expected Fisher information matrix here. Okay, now why am I going on about Fisher information? That has to do with design of experiments, and we will see in a minute why this is happening. Okay, now what is the design? It's usually called Xi, and let me talk you through what the design here involves. So first we've got these TKs, which are vectors of time points for the cave group. And in most medical applications, we would have that all TKs are actually equal. But in other applications, such as in chemical experimentation, we could have different time points for different type of experiments. And then we've got these deltas here. The delta Ks tell us which group we are in. It may just be an indicator function, or it could be something more complicated, like a continuous variable. It could be, for example, the dose of the drug, or it could be even a combination of different doses of different drugs. And in the latter case, if that is a continuous variable, then delta K would also need to be optimized. That's why I've put it in the design as well. And then we also need to know how many patients should be in each of the groups. Okay, now the question is how can we find an optimal design? We have now more or less established what we mean by design, but what is optimal? So if we assume we want to estimate the model parameters in the fixed effects parameter with high precision, then knowing we can estimate them unbiasedly, then we would want to estimate with small variances, essentially. So we could say high precision means something like a small covariance matrix for beta hat, or a large Fisher information matrix, which is why I've gone on about the Fisher information before. But then the problem is we cannot maximize or minimize a matrix. So the idea in optimal design is that we pick some statistically meaningful function that maps the Fisher information matrix into the real numbers. That will make the matrices comparable, and we call this an optimality criterion. An example is de-optimality. And the de-optimal design maximizes the determinant of the information matrix, and that is statistically meaningful because it has the interpretation that at the same time it will minimize the volume of a 1 minus alpha confidence ellipsoid for beta. Hence we are estimating beta with high precision if we can get it to have a small confidence ellipsoid. So in what follows we will use de-optimality, but then there are many other criteria that could be used. For example, for estimating certain functions of beta with high precision, or for accurate prediction. And the choice of optimality criterion will depend on the aim of the study. So I think that chimes in very well with what Kaspar said earlier about first define the aim, and then you can actually think about designing. First find out what do we want to do, and then design the study. Okay, so let's have a look at the results now. Okay, in the toy example, we assume there are two groups, for example, active treatment and placebo, and 4 time points in total. So 1 time point at baseline, 1 at the end, those 2 are fixed, and then we have 2 time points in between, which we want to find the optimized version of. The fixed effects in the model here were just an intercept, a linear term in time, and a group time interaction. That means we will have different slopes in time for the two different groups. For the random effects, we considered 4 cases. The first one is there's no random effect. This is maybe not very realistic in this setting, but still we wanted to see what the design looks like compared with when we have random effects. Then a random intercept model, which is probably the most common one in this setting. And then also a random intercept and slope model where they are uncorrelated and one where they are correlated. We then needed to actually assign values to the variance components, which we've done here, in order to find an optimal design. So these numbers here essentially mean the variance of the random intercept is 1, of the random slope is 3, and this is the covariance between the two. Okay, what else did we put in the model? We additionally allowed some serial correlation to exist in responses from the same patient. And we defined the correlation between responses at times tj and tj dash to be rho to the power of the difference between the tj's, where rho is a number between 0 and 1. So we can see as rho increases, then the correlation increases as well. And also as the time points tj and tj dash get closer together, also the correlation will increase. And for the example, we used a lot of different values, including the case rho equals 0, where there's no serial correlation, and then increasing values of rho. We then also use different dropout functions for the two groups. Both groups start at 100% and end up at 30%. But group 2 has a linear dropout function, and group 1 actually loses patients more quickly, essentially, so the dropout is steeper. Now, I should also say the time seems to go from -1 to +1 here. We are obviously not going back in time with our trial, but we use some standardized version of time. This was initially done to compare our results with results on similar situations that had been published in a paper, and we wanted to see how our designs look compared with those designs. So we are using standardized time from -1 to 1. But once you have a design, you can actually go back to the actual time by a linear transformation, so that there's nothing serious here. And here are the results already. Okay, now this diagram is a bit much, but let's start with the upper panel of the diagram. We can see 4 pictures, and each of them corresponds to one of the models. Now, what is what? We've got, okay, time is on the x-axis, and the y-axis is rho. So that is, and the correlation is going up on the y-axis. Now, what are these strange lines? Essentially, we read this. These are the 2 time points we are looking for. So, for example, I hope you can see my mouse now. When, say, rho is 0.4, and we just move horizontally, we've got a time point here and a time point there. These are our 2 optimal time points for the fixed effects model. Okay, so that's what it means. So one thing we notice is for the fixed effects model, where rho is equal to 0, that is, there's no serial correlation, we see that the time points are at the end. So that design says we should take another measurement at time 0 and at the end of the trial, which is obviously not a very good idea because we are already taking observations at these times. But that is an artifact of the model, which in this case is not realistic because we would not have a fixed effects model without serial correlation in a longitudinal trial, because there has to be some correlation somewhere. Okay, generally, the random intercept model looks very similar, so the design looks very similar to the fixed effects design in all cases except for this rho equal to 0 case. We've got the 2 time points nicely in the center of the time axis, and they seem to be going left a bit when rho increases. For the other 2 models where we have even more correlation by having an extra random slope, then we can see the designs want to take the time points even a bit earlier than just for the random intercept model. Now what else can we see here? Let's go to the lower panel, and this is for the case where we allow the 2 groups to be measured at different time points. And here we remember that the red dashed group had this higher probability of being observed for most of the time axis, and the blue patients were dropping out earlier. And that is nicely reflected here. We can see that the blue lines are further to the left. So these designs want us to take observations earlier on patients who are on the treatment where there's more early dropout. Okay, so that's essentially what's said in this result slide. So for group 1, where we have the lower response rate, the optimal time points are earlier than for group 2. So it seems the design is trying to counteract possible early dropout in this group, which makes sense. We then also looked at proportions of patients in the group, which you could not have seen in the graph. But for all the different cases, the optimal proportion of patients in group 1 was between 46% and 50%. So that is, it's rather close to equal allocation, but it's still, we found it slightly counterintuitive that the group with a lower response rate would want slightly fewer patients in some cases. But that seems to be, it seems to be better for increasing the precision of estimation, but it's not clear why this is happening. Okay, then another observation. I think I mentioned that as well. A larger value of the serial correlation parameter rho will result in taking measurements slightly earlier, as does increased correlation due to more random effects in the model. Okay, so that was our toy example. And now let's move to an almost real example where we try to redesign an Alzheimer's trial that was reported in Howard et al. 2012. And I say we tried it because we made some modifications, and there's still a bit of work in progress because it hasn't worked as we wanted it to work. Okay, in the original trial, there were 5 responses per patient, and the response is the SMMSE score. The time points in the original trial are baseline 6, 18, 30, and 52 weeks. And they were looking at 4 treatment groups: placebo, donepezil, memantine, and a combination of both. And what made the trial interesting to us for trying to redesign was that there was actually differential dropout in the 4 arms. Okay, so now let's have a look at what we actually did, because we redesigned a slightly modified version of the trial. And let's have a look at what happened. Okay, so first of all, we could have just used the trial as is, but then we thought with 4 groups, it might be too similar to the toy example, and we wanted to introduce a continuous variable. So we considered only 2 groups, the placebo and donepezil memantine group, but then assumed that we had some some continuous variable called dose that also needed to be optimized. And then we took the patients from these arms of the trial, and to get a scenario for designing, we then fitted models and tried to find the best parameters which we could put into the design search. So we fitted linear mixed models with various structures for random effects and for the serial correlation. The best model had a random intercept, and it had AR1 serial correlation, as I showed you before, where the parameter rho was approximately 1/3. We then also needed to think of a dropout mechanism, because that also needed these probabilities for dropout. They needed to go into the information matrix for the design. So we fitted a logistic model to the number of observed responses as a function of time and dose. We then used the estimated values from these estimations to plug into our optimality criterion. And we then, for practical reasons, we also restricted the first time point to be at least 6 weeks after baseline, because we thought anything earlier might be too early for the treatment to work. And then we also put an upper bound for the active treatment dose, which was actually the upper bound we used was actually the dose that they had in the trial. Okay, then we found the optimal design that optimizes the time points, the active treatment dose, and the number of patients per group. And now let's see what we got. Okay, let's start with the dose. The optimal dose for the active treatment turned out to be the maximum dose in the range, so that was the dose that had been used in the trial. And when I say optimal dose here, I obviously mean optimal from a statistical point of view, not from a medical point of view. Okay, anyway, so this is essentially what we expected to see. But then the other results were a bit unintuitive. So when we looked at the allocation of patients, we found that of our 144 patients, 84 should go into the treatment group and 60 to placebo, which is similar to what we saw in the toy example, in that the optimal design wants to put more patients in the group with less dropout, but still in the toy example it was still very close to equal allocation. And here it seems rather different. So we were not sure what to make of that, and I think we need to investigate the dropout mechanism. We fit it a bit more. And then the time points. The first time point was 42, so that was the first time point that was allowed. The next one is 285, and then the third one, 356. And again, we are not quite happy with that time point, because obviously the trial ends at 1 year. So it's not practical to get the patients in 8 days before the trial ends. Okay, so I think there's a lot of work still to do, and we need to make the designs more practically applicable for longitudinal studies. So the type of designs we are finding are designs that might work fine if you're just running some chemical reaction and can measure whenever you like, and this is the design that gives you the most information about your chemical reaction. These designs are, the design we found is maybe not very practically applicable in a clinical study. So the question is, what can we do about that? One idea is obviously to put a minimum distance constraint between time points. That is, we would need to elicit expert input in order to see what should be the minimum distance between time points, potentially from a practical point of view of getting the patients into clinics, but also from a medical point of view, what distance makes sense for for this type of treatment. We could also incorporate constraints on the imbalance, because it seemed rather imbalanced what we got, 84 to 60. And in particular, since the 60 were in the group where we actually expect more dropouts, so that was not so intuitive. And in particular, we also need to scrutinize all parts of the model, see if all the fixed effects we have are right, or the random effect structure, the serial correlation structure, and also the dropout model needs more scrutiny. And then in particular, coming back to what Kaspar said before, it's actually what is actually the aim of this trial. And here we consider d-optimality as a criterion because that will give us precise estimation of all the fixed parameters. But we may actually be interested in some parameters more than others. Some contrast might be more interesting to the experimenter, so we could also look into different optimality criteria. How much time do I have?
I think we're supposed to finish at about 3:20, but I think we may have been overrunning slightly as well. So I think a few minutes is still fine.
Okay, maybe I think there are a few slides that I might just keep there, maybe not that interesting. So I'll go to the cost constraint. So this was something we considered in order to establish how many time points we should actually have in the trial. So here for the Alzheimer's example, we incorporated a cost constraint into the optimization problem to find the optimal number of time points. And we assumed that it costs approximately twice as much to recruit a new patient, including taking the measurement at baseline as it does to take a follow-up measurement. In this case, we can then work out how many more or fewer patients we could recruit if we had one fewer or more measurement. And what we did was we said if we had one fewer measurement per patient, then we end up with 172 patients approximately. And we then found that this arrangement was actually slightly more efficient than having these 5 time points for measurements. We compared the optimal designs for 5 time points and 4 time points. And the one for 5 time points had only 91.5 efficiency compared with the optimal design for 4 time points. So this is also something that could be looked into further, because it seems taking, so maybe after sorting out the model a bit more, but it seems that having fewer time points and more new patients in the trial will give us a slightly better estimation efficiency. Okay, and that's my conclusion already. Here we've presented a framework to incorporate dropout into optimal design criteria. And optimal designs can make estimation in multi-arm longitudinal studies more efficient, but a lot more work is needed into how to make optimal designs more practical for medical applications. And I'm saying here where necessary, because if we had a situation like in the toy example, the designs from the toy example actually looked fine to me. And it is the one where we designed the trial where it didn't work out so well. So in that case, we need to make it more practical. So for example, having minimum distance constraints between time points or also bounds on imbalance in the groups. And in particular, we need to look at many further real-world examples, including an unmodified version of the Alzheimer's trial, in order to establish the benefits of optimal designs in this particular application area more generally. Okay, thank you very much. And are there any questions?
Thank you very much, Stefanie, for that talk. Yes, we have, I think, time for a few questions. We'll head off into a break after that, but I think if there are any questions, please feel free to type in the chat, or given we haven't got anything yet in the chat, if someone wants to unmute themselves to ask a question, I think that will be fine while people are typing. So yes, please, yeah.
Yes, thank you. Thank you, Stefanie, for the presentation. I have just one question. It seems to me from what you presented, all the key inputs for this method have to be pre-specified in advance. Is that correct?
Yes, many of the inputs, so the form of the model has to be pre-specified, and some of the parameters.
Okay, so I was thinking whether possibly it's something that you're thinking about to see whether it would be possible to have like an adaptive component which would allow you to alter the solution after you've gathered some data once the trial has started.
Yeah, I guess that would be something to think about. What we have done, the bit I actually skipped, was looking at what happens if we have slightly misspecified the model assumption. So, okay, so I didn't present this slide, but if I just run through this in a second, we looked at how efficient are designs when something has been misspecified. So here we have one design, the first one here, which has a very high relative efficiency. For this one, we had misspecified the parameter rho and the variance component for the random effect. And that design had very high efficiency, so it seemed that our designs were quite robust to parameter misspecifications of correlation parameters. We then had two other designs, which were essentially where we had misspecified the model form of the random effects. They both had a random slope as well, and here they were correlated as well, and that gave approximately 90% efficiency. So what we normally do in design is we look at different scenarios where we might have misspecified our model or parameters, and then see how well would the designs do in that case. Another possibility is also to try to use some Bayesian form where you in a way integrate over certain prior distributions for parameters, for example.
Okay, thank you.
Thank you very much. Simon, I don't know if you're okay to unmute yourself and ask your question, and instead of me reading it out then for some interaction. Is that okay?
Yes. Can you hear me okay?
Yeah, you're fine. Thank you.
So along the same lines as the previous question, I sort of wondered the way you might be able to apply this is take a feasibility study and get the information, as in distributions of dropouts, and then try and use that in the main study. I just wondered if you'd ever actually done that or tried to do that, and how did it go or not go well?
So we are not. So essentially, the studies we are designing are just redesigns of studies that have already been run. So we can always look at the data and then use what we get from the analysis of their data as parameters for our case. But I think it's a good idea generally to look at similar studies, say for example for the dropout, to see there are lots of Alzheimer's trials, and so it should be easy to get information about, for example, dropout in the placebo arm. Okay.
Just my experience is that very few trials look like another trial that's gone before. Maybe that's different in Alzheimer's, but you wouldn't be able to do any of what you've proposed, say, in a grant application for a typical phase 3 trial. It'd be just infeasible. Yeah.
Our final speaker of the session is Thomas Burnett. He's now a lecturer in statistics at the Mathematical Sciences Department at the University of Bath. Prior to that, he was a postdoc at the MRC Biostatistics Unit in Cambridge, and also prior to that, a postdoc at Lancaster University. He has interests in clinical trials, in particular adaptive clinical trials, and he will be talking on the missing data problem in adaptive clinical trials in this talk. So I'll pass it over to Tom now.
Perfect. Thank you very much, Robin, and thank you for the invitation to speak today. It's a privilege to be joining you guys. So as Robin said, I'll be talking on the missing data problem in adaptive clinical trials. So just to give you a quick outline of the talk, for those of you that have seen me talk before, I do apologise. I'm going to start with an introduction to what are adaptive designs, try and convince you that they're a good idea before telling you about the problems with them. But hopefully that will put us all on the same page in terms of what it is we're trying to achieve. We'll then talk about where the missing data comes from specific to adaptive designs. So this is slightly different to missing data that we might consider more generally, but as a consequence of the design. I'm going to introduce some hypothesis testing frameworks. This is more so that we make sure that we have some kind of aim for what we're trying to achieve. We'll discuss it as we go, but it's obviously not the only important outcome for a trial, and your mileage may vary in terms of actually what your important outcome is. We'll talk about decision making, and thus that will lead us into how the missing data problem essentially can impact that decision making and thus impact the designs overall. And we'll finish up with some discussion. Now, I plan to point it out later on where it becomes relevant, but I should note that this is kind of contributory to ongoing work with Robin, who kindly introduced the talk. So I'm sure I don't need to introduce him further, but I'll point out later on where that work is going. So hopefully you can kind of get an impression of what we're up to. So first up, what are adaptive designs? Well, the crucial idea is that we're going to modify an ongoing trial. And today we're going to focus on the idea of doing this by design, i.e. in a pre-planned way, based on the accruing data available at the interim analysis. And the idea is that this will enhance the flexibility of the trial from the design point of view, without undermining the integrity or validity of the study in question. So that's kind of the core idea. Just to kind of give a little bit more context, we'll compare what this means in comparison to what would be a more traditional fixed sampling design. So in a fixed sampling design, you might plan your study and fix your sample size at the beginning. You then start recruiting patients, and essentially the idea is that on this timeline, you continue recruiting patients until they're all recruited, and then you conduct some final analysis based on all of the data available from the trial. So some kind of short notes on this concept. The total sample size in this case would be known in advance, but there's no adjustment possible. So should the data that accrue as we go along kind of conflict with some kind of element of the design, we're not able to make an adjustment based on that. And this is where that idea of adaptive designs comes in. So in addition to our initial planning of the study and fixing of the design, it might seem weird that I'm calling this a fixing of the design, but this comes back to the pre-planned element, so the decisions that we make along the way. Today we're focusing on those as pre-planned decisions, so they're still part of the design of the study as far as I'm considering it. So we then start the study, but in between the kind of start of patient recruitment and the final analysis, you'll notice this idea of interim analyses has appeared. And so at each of these interim analyses, we might do one of several things, depending on the design in question. We might do one or even more of these things. So we could, for example, decide whether or not to stop just for futility. That's the kind of main example that we'll use today. We could change the sample size. We could drop or add a dose to the scheme. So we looked at multi-arm trials in the talk just before. We could change the endpoint, or we could change the question itself in some pre-planned way. So some pros and cons in general. So before we go into the specifics of the specific con, essentially, that we're going to be looking at for the majority of today's talk. These designs are highly flexible. That's both a pro and a con, actually. So the high flexibility is useful in terms of when we're able to make good decisions and change the conduct of the trial in a useful way as we go. But that flexibility is also a problem from a design perspective and from an admin perspective, in that we have to account for these possible pathways through the trial. They can be very efficient. They can also be inefficient. And so this is the concept. We'll talk a little bit later on about group sequential designs that you're most likely to be familiar with, of the things that we'll mention today. These have a lower expected sample size, but a higher potential maximum sample size if you compare them to what you could have done if you just fixed the design, or used a fixed sampling design, I should say, from the outset. The other pros are maybe a little bit more dependent on using them in the correct way. So they may reflect medical practice better, depending on the adaptation you use. You may end up with a shorter trial and or more accurate estimates. Often you'll find that you're trading off those things, though. They might be more ethical. So from the perspective of, if we think of the multi-arm designs that were talked about earlier, if you're able to remove arms that you are able to show early on they're not effective for patients, that's an ethical benefit. On the counter side, obviously we've mentioned they're time consuming from a design perspective. Post-trial estimation becomes more difficult, and maybe that's something for the panel discussion in terms of how that ties in even further with estimands. Simple estimates may be biased, and so this has been recognised in guidance on adaptive designs of having to understand the bias. And the interim analyses, certainly as we'll view them today, we're viewing them in an unblinded way, which may not be desirable. So just a very quick kind of whistle-stop tour of some common adaptive designs, just to illustrate that they are prevalent throughout or available throughout the clinical development process. Prevalent is probably not the correct word, although they are gaining in popularity. So I won't go through these in detail, but we've kind of mentioned a couple of these, and you might see others that you know, and we can talk about those if you have questions later on. The key point is there's some common concepts that unify these designs, and those are the common concepts that we're going to be talking about today when it comes to how does missing data impact the design. So specifically, we're looking at missing data that is caused by the design itself. But let's take a step back and think about recruitment to the trial in general. So not all patients will arrive to the trial at once, and adaptive designs are going to make the decision based on accruing data. So you can see our picture here of a simple two-stage design. We collect our data from stage 1, and then that's going to inform, well, how should we recruit stage 2 of the design, broadly speaking. So the decision for the design, or which part of the design we'll be using for cohort 2 here, will be based on these available data from cohort 1. Just an idea of some possible decisions. So just some very simple ones for today, but based on our observations, we might, for example, continue the trial as we had recruited stage 1. So here it's just represented as the same number of patients, for example. We might increase the sample size. So perhaps the treatment effect is looking reasonable, or the estimate, sorry, of the treatment effect is looking reasonable, but we might need a little bit more data to push us over the line. So we might increase the sample size, or, and this is where we'll come back to with our example, we might stop for futility. So we might say the estimate of the treatment effect is not sufficiently big at this stage. We're going to stop the trial early for futility. Well, now let's think more in depth about that recruitment to cohort 1, upon which we're going to base our cohort 2, or the decision for how we recruit cohort 2. And we'll know, particularly with the talks earlier, that patient outcomes are not going to be immediately available. And so given this kind of rolling nature of recruitment, we're inevitably going to have some situation where if we cut the trial at any point in time, we may have recruited some patients for whom on the timeline we've kind of got to their final observation, their final endpoint, but there's also going to be some patients who will be observed at some point in the future. And this is where I'm kind of claiming that by design, we have our missing data problem. So if we imagine this dashed line as our interim analysis, And you can see by the kind of the grayed out side of the timelines in the future, and so currently not observed. So if we take a really simple example, suppose outcomes take 3 months to observe and the patients have been recruited kind of constantly over 6 months, which may be an oversimplification as trials kind of ramp up as they get going, but it will suit us for now. So at the 6 month point, we'll only have seen observations for the first half of that cohort, i.e. those recruited in the first 3 months. And so what should we do? How should we make our interim decision? Now, at first, I'm actually going to present an option that doesn't have a missing data problem, but is hopefully intuitively slightly unappealing. So we could start recruiting the next stage of our trial based on our best guess. So what at the start of the trial did we think was the most likely decision that we would make at the interim analysis? Let's continue recruiting based on that, under the assumption that we can't just stop and then restart recruitment. Hopefully that seems like an intuitively bad idea for anyone listening on that one. So we compute the interim decision then once all data are available. So we can carry on recruiting cohort 2. We wait until we've observed cohort 1 and we decide what to do with the remainder of cohort 2 at this stage. Okay. However, in this kind of really simple example, hopefully you can see it's possible that we end up with a decision that's in conflict with what we've actually done. So if we hop back a couple of steps, we had this option of stopping for futility. Well, if we stopped for futility, is it really that ethical that we continued recruiting patients while this was a possibility? Let's suppose it's not, and park the discussion of whether that's a reasonable thing to do for now, and look at option 2. So option 2 is to use the available information that we have, i.e. the patients that are both recruited and observed, and make the decision at the interim analysis. So this decision is essentially made with missing data on the other patients that we've recruited but not observed. Worth noting, all of these patients from cohort 1 will still contribute observations to a final analysis where we may be making our kind of final decisions. So in this example, our interim isn't making a kind of stop for efficacy decision like you might see in group sequential trials, and so that's okay. And the larger the delay, obviously, the more patients are missing. And the real question that I want to try and answer today is, how does this impact the trial? So while it's kind of a known problem, it's maybe not made so clear outside of the adaptive designs community. So as I said, we're going to use hypothesis testing as our real measure of performance of the trial. So in particular, let me just kind of point you to the key points. So we're going to define a null and an alternative hypothesis, and we should test this. This is supposed to say at level alpha, but hopefully you can kind of follow along with the usual idea of hypothesis testing. And so each of these theta might be a treatment effect, so a difference between the control treatment and the experimental treatment, for example. And we'll see an example of that later on. I mentioned group sequential testing, so I would be remiss not to kind of put up the usual picture of this. And so this is the idea that if we have some test statistic, then at the interim analysis, we can make our conclusion for efficacy or continue recruitment or fail to demonstrate efficacy, i.e. futility, as we've been calling it, and then continue to a final analysis. But this is a formal hypothesis testing step in the interim analysis. And so there are other alternatives if we don't wish to take this step. In particular, we're going to focus on the concept of combination tests. So supposing we have some p-values from the first and second stage of the trial, p1 and p2, the idea is that we can construct a overall p-value for the trial that is also uniform 0,1 under the null hypothesis. And this p-value will be able to be used for our overall hypothesis test. The important thing to note here is that this construction essentially allows us flexibility over how we make interim decisions. So by construction of these combination functions, we can make the type 1 error be exactly controlled as we desire. And that gives us freedom to choose from our pre-planned set of decisions. Recall from earlier, we had 3 possible decisions. And so we construct our hypothesis testing routine such that no matter which decision is made, the type 1 error is controlled. And that's the key point for many adaptive designs and the decision making. Now, this will tie in later on with the discussion, but it's not so crucial for now. But I just wanted to introduce the concept of multiplicity. So supposing we have instead 2 experimental treatments, for example, we may now have 2 sets of hypotheses, and we might be requiring, for example, strong control of the family-wise error rate, so a stricter level of control. And in addition, this creates more complexity in terms of how we make decisions. So we'll come back and touch on this kind of concept later on, but it's not going to be the focus for the rest of the talk. Worth noting, we can still do everything that we claimed we could do in the case of a single experimental treatment. So any procedure that controls the family-wise error rate can be written as a closed testing procedure. And within the closed testing procedure, essentially what we can do is apply a combination test within each relevant hypothesis for the test. And this will give us control of the family-wise error at level alpha by following these global rejection rules. So if we wanted to reject, for example, if we have H01 and H02 as our 2 null hypotheses, if we want to reject H01 globally at level alpha, we must also reject level alpha tests of H01 as a local test and also of the intersection H01, H02. And this can be extended. So if you have more hypotheses, you essentially look at all possible intersections of hypotheses to ensure you're testing. And as a very quick note, but this will hopefully give some clarity later on, if we are thinking about how to test these intersection hypotheses, this is a very simple example of what that rule might look like. So suppose we have Z values on each axis for each treatment group, or experimental treatment group 1 and 2. Then the dashed lines are essentially our local tests of H01 and H02, so in the usual way. And our kind of green line here is a region on the outside of which we construct at level alpha following a Burnett type test to test for the intersection hypothesis. The point of showing this is that you can see there's now an interaction between the experimental treatment groups. Essentially, this comes about through the common control. Okay, so just moving on to the decision making within adaptive designs. So just to kind of formalize that point that we can construct this decision rule however we like, essentially what we have is that our decision rule must be some function that outputs the possible decisions that we could make at the interim analysis. But my claim is that we can choose how we construct this decision procedure, essentially, so long as our testing procedure is valid no matter which choice we make. So just to re-emphasize that point. What this boils down to in most cases, and certainly for our discussion today in one dimension, is essentially a decision threshold. So this applies in many adaptive designs, not all, and we'll hopefully have time to discuss when that's not the case, particularly in multi-parameter problems that are in some sense optimal. But the idea is that we could have some threshold. So if we think back to our decisions before, if we have a theta hat 1 from our observations from the first stage of the trial, we might continue the trial if that theta hat 1 passes some threshold A. We might increase the sample size if it passes some threshold B. And this should be a B in this final step. We might stop the trial. So this is our futility stopping if the estimate of the treatment effect at the interim analysis is not sufficiently strong. So very simple thresholding idea. And we're going to essentially use this thresholding idea to illustrate the potential impact of this missing data. So we'll now move into a bit more of a concrete example. So a very simple example. For those of you that know adaptive designs, you'll know we could get much more complicated than this, but we'll follow largely the construction we've looked at so far in this talk. So supposing we wish to test the treatment effect and a null hypothesis that it's less than or equal to 0 versus an alternative that it's greater than 0 at size alpha. So we'll just take that as 0.05 for today, where our theta is the difference between our experimental treatment and our control treatment. Well, we might plan a two-stage trial, and we're going to leave open the option to stop this trial for futility. So that's going to be our adaptation. We'll come back to how we make that adaptive decision momentarily. But essentially, for today, we'll just assume that these observations from patients are going to be normal. And what I've essentially assumed is that we have the same sample size on treatment and on control and within each stage of the trial. So we're stopping halfway through to make our adaptive decision. Okay, important to note, this is for the end of the trial. We will construct our estimates theta hat 1 and theta hat 2 based on the cohorts of patients recruited before and after that interim decision point. So kind of coming back to our recruitment idea from earlier. Now, I didn't mention this when we were talking about combination tests, but the one that is our kind of typical go-to, because it essentially is most powerful when the weights are correctly defined, is the weighted inverse normal. So we're going to combine the Z statistics from each stage of the trial to give us some overall Z in the same way that we would find our overall P. So this Z will be distributed normal 0, 1 under the null hypothesis. Okay. And a note just for this example, I've chosen n and sigma squared such that essentially we end up with a power of 0.9 for a treatment effect of theta equal to 10. So just as a reference point for what's to come. Obviously, we could scale n and sigma squared to whatever we wanted, but this is just kind of a simplification that will tell us everything we need for today. So what happens at the interim analysis if we, sorry, I've skipped the slide on where the missingness comes from. Oh, no, I haven't, here it is. So at the interim analysis, we assume that a proportion of the data from cohort 1 is missing. So we'll call this tau. How does this impact the variance of our estimator? So instead of having theta hat 1, we'll now base our decision on theta hat I. And we can see if there were no missing data, essentially the variance is the same value as it would have been if we'd had no missing data. But we can see as we decrease the proportion of data that's observed at the interim analysis, we increase the variance of our estimate. And this is what has the impact on our decision making that we'll illustrate in a moment. So you can see down to about 0.5, it's not too bad. It's not a huge inflation. But then as we start to head into this region below 0.5, the inflation is going to be much, much larger. Inflation of the variance of the estimate that we base our decisions on. Okay, and so in this case, we're going to base our, as I argued in option 2, we're going to base our decision on the available interim observations, which will give us our theta I. And we're going to stop for futility. The inequality is the wrong way around here, but based on a simple threshold. So if theta hat I is less than some A, then we're going to stop for the trial for futility. And otherwise, we continue recruiting as we'd planned. So again, equal recruitment, treatment and control, and to each stage of the trial. So let's take a specific value and think about how this would impact the trial and how the missingness would impact the trial. So let's suppose if our treatment effect or our estimate of our treatment effect was 0 at the interim analysis, i.e. we're not showing that there's any benefit at the interim analysis, how's that going to operate as a trial overall? And so here in this table, what we have is the probability that we stop for futility, and then the probability of essentially what we defined as our successful trial rejecting the null hypothesis. So if there were no missing data, we wouldn't stop for futility. Sorry, this is written the wrong way around. If there were missing data, there would be the opportunity. No missing data, there would still be the opportunity to stop for futility. What this column actually represents is the design of the trial if we didn't have futility stopping. Okay, but if a quarter of the data is missing at the interim, we can see we stop 15% of the time with about 80% power. If half of the data is missing at the interim, we then stop less often. Recall this is under the alternate, so theta equal 10, where we're comparing this to a stopping boundary of 0. And so you can see that as we increase our proportion of missing data, we're slowly able to make a better decision at the interim analysis. And essentially what we do is recover the probability of rejecting the null hypothesis that we would have had with a non-adaptive design. So to show that again, but more broadly, so looking over a case of what happens as we change the delay. So changing the delay on the x-axis, plotted against the probability of futility stopping on the y, and over on the right, we have the probability of rejecting the null hypothesis. So we can see again, it's essentially this 0.05 to 1. We're making broadly the same decisions as we would have been. And then if we have more missing data than that, we step into this region where it's harder to make good decisions. And you can see in the extreme, we start to lose a reasonable chunk of our power to reject the null hypothesis. Okay, so you might be thinking, well, that's all well and good, but if I know my delay, I might be able to design things with that in mind. So I could change the design and potentially fix the probability of futility stopping, as it were. So essentially changing that threshold depending on how much missing data you're expecting there to be. So let's have a look at what happens in that case. Well, if you look on the left, this is under the alternate, so theta equal 10, the probability of rejecting H0. We can see actually this is pretty good, no matter how much missingness there is. If we fix our probability of stopping for futility at 10%, then, or at least under the alternate, then there's not a huge difference in power. So we go from 0.81 to 0.85 just across the range of amount of missingness that you might have. The problem in this comes really in this upper left region of the second plot. So this is under the null hypothesis, where we really would want to be stopping for futility. And you can see there's a huge discrepancy depending on how much data is missing as to how often we manage to stop for futility in a case where if we really believed in futility or our futility boundary that we had initially, we really should be stopping. So, for example, this should be kind of a fixed 50% under the null hypothesis, just given that the futility boundary is set at 0, which is the null. So you can see, again, in this lower region, we've still got some problem. And so it's not a simple solution when you're balancing these characteristics against each other. So just to conclude, we'll move into just a little bit of discussion. Hopefully I've illustrated that this is kind of a wide-ranging problem and given an impression of where it might come from. Essentially, there are several implications of this missing data. And so the first and the major one is that it can change the behavior of our interim decision from what we had expected it to be, or what we may have designed it to be. The overall probability of rejection is equally influenced by these interim decisions. And so we now end up in this place where we've got a trade-off. We've only seen one kind of binary decision and one binary result, but both of the above are further influenced if you had multiple endpoints of interest. So that multiple testing scenario that we talked about instantly makes this a more complicated procedure to really understand what is the impact of my missingness going to be. And then decision rules that aren't simple thresholds. So as we saw in the hypothesis testing, we had this kind of stepped function that was dictating the hypothesis test for the intersection hypothesis. If you've got multiple endpoints or multiple things that you're concerned with as outcomes of the trial, you may no longer have a simple threshold problem. And so it's not as simple as saying we want to fix this probability, because your threshold may not be kind of essentially what we have here is a flat line, but your threshold may interact between your multiple endpoints or your multiple outcomes of the trial. And this then impacts overall optimization. So another big topic for me in my research is how do we optimally design these trials? We've seen kind of some discussion of optimal design earlier today, and that's going to be heavily influenced by, in this adaptive setting, how we make that interim decision. And thus it's going to also be heavily influenced by the missing data. So overall, this might limit adaptive designs, or in fact, it does limit adaptive designs to settings where data are relatively quickly observed compared to the planned trial length. So you can think of the extremes. If you have all of your data available at the interim analysis, that's pretty much the best case for an adaptive design. If you have no data available at the interim analysis, this would be the worst case, because you're not really able to make a different decision to that which you made at the start of the trial. And so there's really no point in the adaptation unless you do it based on external factors. We'll come to that in the next slide. So there are some potential solutions to this problem, and it really kind of depends on the specific adaptive design as to how much work has been done on this. For example, in group sequential designs, there is work on what you do with these pipeline patients and how that relates to the stopping rules. For flexible methods, we have more freedom over how to make these decisions. So we could, for example, consider the surrogate endpoints. Kaspar talked a lot about estimands. Maybe use a different estimand to decide how you're going to make your adaptive decision as to how you're going to make your decision at the end of the trial, and think about how that's related. We saw longitudinal endpoints in talk 2 from Stefanie. So that joint modeling of endpoints could be another way to kind of recover some information. If you have something that is less missing with your more missing data, your endpoint with more missing data, you may be able to jointly model this and have a better understanding. Or you may have some external drivers to these decisions. So what's going on in the wider landscape of treatment for the illness. And then just to point out, this is where the ongoing work comes in. And so the idea here is to then start looking at using missing data methods to better inform these decisions in the case when you've got missing data. So really kind of directly tackling the problem, as opposed to finding different solutions that are maybe not exactly what you were trying to do. And so this just leads me to the summary of my talk. So hopefully today I've given you the impression that adaptive designs can offer more flexible, efficient and ethical trials, but there's no free lunch. So you pay a price for this in terms of the extras in terms of design, and some penalties in terms of potential efficiency. So you really should be using them where you think they're actually useful for the specific design. As part of the design, we then may have to make decisions with missing data present, and we've seen hopefully how this might impact the overall operating characteristics. So we saw one very simple way of coming around to a solution. We can see that actually this is more of a design problem than might be kind of apparent at a surface level, so it might be something that needs further consideration. The implications of what we've talked about today, as I've said, are much further reaching in more complex designs. And so as you start to get these interactions building up, it makes these decisions harder to make, and thus the missing data has a bigger impact in that setting. And while some work does exist, it varies from design to design. And so our hope is that directly applying missing data methods may offer some kind of more generally applicable solutions to these problems. That's it from me. So I will hand back to Robin now. I'm sure there'll be some time for questions and then the panel discussion. Thank you.
Thank you very much, Tom. So yeah, I just put a message in the chat to say if there are any questions, please yes, great, fantastic. So so I'll just bring Michael in, if that's okay. Michael, do you want to just ask your question, if that's okay?
Yeah, thanks, Thomas, for a really lovely talk. I was just thinking about in the situations where your endpoints might be timed to event, and whether you've considered that in your work, where there might be partial information available in terms of like a censored outcome.
Yeah, so this is a great point, and again, that has its own pros and cons. So with the censored outcome, you may have kind of be able to derive some understanding from the idea that it's censored, but also we didn't talk about other potential sources of missing information here. And so if you have other sources of missing information, such as you might have in time to event analysis, then this is going to make the decision making harder as well. And so you already see how, one simple change, such as going from just normally distributed observations to time to event, makes what we presented today, which was a very simplified version of reality, a much harder prospect to think about. Okay, but how do I kind of account for the interaction of these things? But absolutely, it's something we will be considering. It's a great point, because it's an important element of trials. So it's an important element to be able to offer efficient designs in that case as well. I guess it's worth mentioning that time to event is probably one of those endpoints that is more likely to be not so useful for an adaptive design, because you inherently have this delay between recruitment and observation of the information that you really need to make those decisions. So again, it illustrates that kind of difficulty of, is an adaptive design able to deal with this structure of data?
Thank you. Yes, I've come across them in dose escalation kind of designs before, but it's not really the hypothesis testing area that you're talking about. But yeah, thanks very much.
Yeah. I guess just to mention on the hypothesis testing, so that was, again, just an illustrative example, but as Michael's pointed out, there are many other areas where actually hypothesis testing isn't the core thing that you're trying to do. And so that's another relevant point in terms of the nuances of, as you go into specific areas and specific designs, of what you're actually trying to achieve matter quite a lot in terms of this interaction with the missingness.
Thank you very much. Yeah, no, that's a really interesting point on design for censored observations. I know that one of our other speakers, Stefanie, has done some work on that as well. So there's some really interesting work there. I guess we should move to the next question, because it's so. Richard, did you want to come in and ask your question?
Yeah. Excuse me. Hi, Tom.
Hello.
Yeah, I was just wondering. You made the point that adaptive designs may not be that useful in situations where you have to wait a while for the outcome to be observed. And that's one of the challenges we've had in using adaptive designs in mental health, where it's not a short-term outcome that's often considered. So what I was wondering is there a kind of sweet spot, a trade-off between the accrual rate into a trial and the length of time between that baseline and the primary endpoint? Because that will influence both the amount of missing data you're going to see, which is the problem you tackled, but also just whether the use of an adaptive design is therefore suitable as a function of those two things.
Yeah, and this is a fantastic point. So we kind of glossed over this entirely in the talk in the interests of time, but the accrual rate and then the overall time of the trial and the length of time that you're going to observe each patient is all in the mix when you're thinking about, okay, how much missing data will I end up with at the interim analysis? And so this is one of the things that you then need to be considering when designing these trials. So if you had, for example, a very long endpoint to observe, but your accrual rate was quite slow and you planned for the trial to be over a number of years, well, actually, in that context, you might still be able to use an adaptive design, because of that mechanism of, as patients come in, we'll still have enough observations to make some informed decision a couple of years into the trial. And so it's all relative. There's no kind of on paper answer I can give you on this one. I think it's part of what we're kind of looking at here, is to give the tools to start making those evaluations and start having that understanding. But very early stages of that work. But it's a fantastic point. So thank you for bringing it up.
Great, thank you very much. So I don't see any more questions in the chat. and it's actually perfect timing because it's 4:30, so which is when we start our panel discussion. But if there are any questions, that's still fine, of course, to be asked here. But also, we can also move into sort of general questions for any of the speakers, but also broader sort of questions with respect to design and clinical in the clinical area. I think so I think what we'll do is, again, similar to before, really, if people want to just type their question in, and then I can just, so I have an ordering of, or raise hands, that's also fine, and I can just bring people in as possible. So Simon has raised his hand, so I'll ask Simon to unmute himself and I guess ask his question, if that's okay.
Okay, hi, thanks very much. Yeah, I'm Simon Day. I actually wanted to ask a question to Kaspar, really, about this Estimand stuff. I remember a few years ago Frank Brett saying that it was often the statisticians who actually decided what the question was that we're going to answer in a clinical trial, because it got left to the statisticians to decide how you're going to analyse it, how you're going to impute missing data, and all of these things effectively mess around with exactly what question you're asking. And as Frank was saying, yeah, this is a pretty absurd situation where you have the statisticians decide what the question is. Has anything changed? That's my question to you, Kaspar. I mean, I know this is supposed to change things, and you said in your kind of closing slide it requires a multidisciplinary approach. Is there any statistician who thinks designing clinical trials doesn't need a multidisciplinary approach? It should always have been there anyway. But do the clinicians, at least, well, the non-statisticians in general, really engage in this Estimand thing? It seems to me that they won't, and it kind of gets left to us to figure out what the intercurrent events are and what we're going to do with them. And I'm just not actually convinced that we've solved this problem of the clinicians really not pinning down exactly what question they want to ask, but they're kind of leaving it to us. My question is, what's your experience of that? Are you having complete success or not?
So yeah, thanks, Simon, for the question, and you of course call out a very important point. Maybe let me start with saying, I agree, statisticians often decide the question, but even they do it implicitly. For example, with all the methods we use for missing data, they might imply one or the other estimand, and sometimes it's not even clear, and we just deal with it somehow through the estimator, and we never really loop back and say, okay, if we use this estimator using some methods for missing data, is this actually answering the question we set out to answer? Now, you're asking about my experience. I think in the large companies, there is a huge effort going on implementing this Estimand's addendum, like with company-wide initiatives, with trainings for everybody involved, and also using striking examples where it becomes clear to everyone, if we had this discussion at the onset of the trial, things would have been very different. And with this, you can bring the clinicians on board. And even so, we organized PBS seminars in Basel together with clinicians. We start to publish together with clinicians, basically emphasizing that you need to be clear about the potential patient journey through a trial, and then that will help you define the intercurrent events. And then for every intercurrent event, you need to define a strategy. And I think this is a great opportunity for statisticians and quantitative people, because they can help lead the team through that. But we need to assume that leadership role and point these weaknesses out. I think in the past, even ourselves, I mean, we had a trial, we analyzed it, we had 1,000 patients, we implemented the primary endpoint, and then we went back to the team and said, for 940 patients, I have implemented the endpoint. Here is a list with 60 for which things are not so clear. And then they kind of went through every patient and decided what to do with the available data for that patient. And that's not what should happen. I think then we need to have these discussions much earlier. And slowly, I think we bring clinicians and stakeholders on board, because they also see the benefit. but you need to work hard to showcase that benefit.
If I just a couple of comments. Well, first comment is I think I agree with everything that you've said. But I would just point out, you say, in large companies we're doing all this sort of stuff. What about the thousands and thousands and thousands of small biotech companies out there?
Yes.
Who actually do most of clinical research? The Roches and the Pfizers and whatever, they only do a minimal part of it. Most of it is done by the thousands of other companies. So I see a problem there. And just one other comment, which you can laugh at, but you don't have to answer. I was recently in a meeting with a certain regulatory agency. It's the one that's abbreviated the Finally Decisive Answer, the FDA. And we were arguing essentially about what the S-demand should be. And the division director said, OK, we're not going to sort this out by phone. Write it down, send it in, and the statisticians will get back to you. And I thought, no, this is completely wrong. But so the FDA think it's a statistics problem as well, or at least that division director did. But anyway, thanks very much for your comments. And
I can maybe reply to the two comments. And again, you're perfectly right with the biotechs. They have less resources to spend on that. But I think they start to feel it. They get pushback from regulators. And we've built this oncology S-demand working group. You have similar working groups in related areas. We give a lot of trainings, webinars. We try to help this. We start with the stats community, but we are also kind of broadening out to clinical community. I think ICH 9 guidelines or ICH guidelines, they're usually understood as some kind of high-level principles. And it's always the case. You need to make this concrete. What does it mean for a trial? And there is some work involved. And I think we try hard to provide that work. So that's one thing. The other thing is, as far as I know, the working group that wrote the addendum is currently discussing with journals to bring them on board, so that when you submit the trial to JCO, New England Journal of Medicine, Lancet, that I guess it will be buried somewhere in the supplementary material, but that as today, you need the endpoint defined, the sample size computation, you might need to have the S-demand properly defined at the design stage. So that's another thing. And then FDA, my experience is that initially there was pushback from FDA. And now at least the statisticians are really jumping on that train. For example, if you look at the ASA FDA workshop this year, and we're currently planning sessions for next year, a lot of FDA statisticians are reaching out. Can you come? Can you present? Can you provide examples? It's like in the companies, it might take a while until it has reached the clinicians as well, and that we can fight that perception. It's a stats thing, because that's also what we have to do, work very hard in our company to not have this pushed to the statistician. And then FDA, my last word I would say is, we often talk of the FDA, but for me, FDA is a collection of reviewers. So that's very heterogeneous, and you might end up with a reviewer that is more on board with it, depending on who you get. Yeah. But thanks for the questions, very pertinent.
Can I ask a follow-up to that, Robin? I was just wondering if I could jump in and hassle Kaspar some more. Essentially, what you're looking at, and it's been a long time coming, and I've kind of been observing from the side as a methodologist in academia, but it's obviously a cultural shift in terms of the way that we tackle things and essentially the way we go about conducting ourselves. And you talked in your answer there about the training that the group offer, and I was wondering, maybe this is just my personal viewpoint on it, but has there been much, do you know if there's been much uptake of teaching this as a concept at university level, such that the kind of pipeline of people who are coming through are being trained in it before they even get to the stage of the industry side? Is that something that is in place, or is it something that's coming, or is it something to consider in the future?
Something that is helping with this a lot is the rise of causal inference also in stats degrees. I mean, this is all very related, although the ICHE9 addendum has a little bit of special view on these things. But when people are familiar with what potential outcomes are and kind of how you apply these causal inference principles to observational data, I think they have an easy access to this whole estimands discussion, although things are a little bit different. The same applies for people who are trained in epidemiology. When they are exposed to the target trial framework, I think then it's a small step to understand what is this whole estimand thing all about. And putting the question front and center and not jump on, we are going to use a mixed model to analyze this data. That's technical details which you can leave to the statistician. You need to first define what is the question. And my experience is with younger people, given their exposure to causal inference and at many universities, meanwhile, when you do a stats degree, they have access to this kind of thinking. But from our side, we offer webinars, but that's for pharma industry. I mean, via DAA, via biometric, the International Biometric Society. Not sure how much we reach students with that, so it's a fair point that there is no kinds of concerted effort to bring that to universities. Yeah.
Great, thank you. I don't know if anyone else if any other comments panel want to say on this. Feel free. I'm just gonna, as we're on this topic, Susie's not posted a question, but she's just shared, as people will have seen, some information on the chat about this MRC NIHR trials methodology, and they're going to be running a workshop on this related area. So it's great to, people are thinking about this, and it's very interesting to know about that, just because as in I wonder sort of what the motivation is or what the reason is that that things are left to the statistician to decide, as opposed to sort of having a collaborative sort of or some sort of joint thing. I suppose there's some investment in sort of trying to think through the problems as well, maybe. But I mean, please feel free to put questions in the chat. But I don't know if this is sort of slightly related but tangential to it as well. Is the question I had looking at some of these talks and seeing some of the discussions we're having. There's a lot of non-maths or statistical side issues related to clinical trials or design, right? So like from what when Stefanie was sharing, when it was presenting, we saw that there was sort of for example two time points very close to each other, or and actually there was an interesting thing that I've heard of in other talks, when in Stefanie's talk as well, there was the placebo arm had fewer measurements than the treatment arm, which was counterintuitive to the process. But if you were to argue the other way around, and you had more in the placebo arm than the treatment arm, you would get people saying, oh, well, that'll never fly. You can't have put more people in a placebo than a treatment, because what, especially if it's very much different. So, and coming back to Tom's point about ethics, and in his talk as well about, like sort of adaptive designs. And so at what sense how can that feed, how do you weight those relative importance, and how do you put a, how do you kind of factor those sort of things in? I mean, is there a way to do it in a principled way, or is it going to just essentially have some sort of heuristics or sort of non-sort-of quantitative ways to deal with that?
So I guess I can maybe give some insight from the adaptive design side to kind of kick us off. And again, this comes back to the discussion that we had while I was talking and afterwards, of what is it that's your kind of goal of your adaptive design. And so, for example, I think we talk, or at least in the list of kind of commonly used adaptive designs, response adaptive randomization was one of those in that list. And that's a setting in where there is a lot more work done in terms of, okay, are we looking at a balance of ethics or output, or what is it we're trying to achieve? Equally, adaptive designs can be quite useful in small populations. And so you actually start to think of potentially your goal of your trial slightly differently, in that you're going to be treating a large portion of the population rather than trying to figure out what the drug is. Your goal is almost, as we go through the trial, we should be giving as many of these people the right treatment as possible. And so again, it's kind of that response adaptive idea of what is your goal of the trial, and how do you weight that together, and I guess some kind of utility type way for then optimization, which may be a point for the others to jump on from.
Yes, I think it would be some kind of multi-objective optimization. So you really need to talk to clinicians and those who will actually run the trial, and find the estimate, obviously, and then know what the estimate is. And it's really a collaborative approach to see what is needed, and then you will have some kind of multi-objective optimization problem. You need some utility for finding the best case.
Maybe I can comment on that also from a pharma perspective. I'd like to make 2 points. I always felt that the main, although that's not perceived as such, but the main contribution of a statistician in a development team is not the statistical knowledge. It's the ability to phrase the question, to structure the question, and then kind of, that's what you learn as a mathematician. You have this huge problem, you have no clue how to solve, and then you chop it up in small pieces, you solve every little piece, and then you plug it together, and then you have solved the big problem. And I think this mindset and this ability that we are trained in is very, very helpful. And often, I mean, of course, you can define a utility function, but often there are many things that you cannot quantify. And then you need experience, you need kind of this analytical thinking. And I think that's, for me, the process of when you enter pharmaceutical industry, you have a stats degree, you enter as a statistician. And when you retire or when you leave, then you are a drug developer. And that's exactly the difference between these two things. You learn the methods, but then there's this huge universe of developing drugs. And if you, when you work a lot in these projects with a lot of molecules and with a lot of people, you gain this knowledge and seniority and become a drug developer. So I consider myself a drug developer, hopefully, meanwhile, and not just a statistician anymore.
Yeah, no, I really like that way of thinking about it, because it's so often that's the main challenge, to convert a practical problem into something that can be solved with the statistical theory and knowledge that we've learnt about. It's one thing pushing a button in SAS or something, or SPSS to get an answer, but figuring out what the question is often, at least in my experience, a lot of the work. So Richard, you posted some interesting thoughts there. Did you want to elaborate or come in with that?
Sure. So it's a really great session. It's really stimulated me to think through some things. Playing devil's advocate about the Addendum framework, one of the nice things we've always had is the intention to treat analysis is kind of clear, that you analyse people in the groups they're randomised, and that's always been a kind of fallback, that we can just say, no, it's not ITT, we're not going to do it. And when we've been asked to do additional analyses that we may think is less principled, my slight concern with the Biedermann framework is it opens the door just a centimetre to make some sort of post-randomisation exclusions, or to censor people after an intercurrent event and so on. And for less scrupulous trialists, if you open that door a centimetre, it could blow open. And there's a danger that lots of trials where they're not sort of overseen in the degree of rigour that Kaspar's explained, in pharma and in his group, that this could just be abused. So I'm just wondering, and I can see that might be the appeal of the Biedermann framework for some of the clinicians, to pick up on Simon's point earlier. So I'm just wondering how we sort of protect against that across all clinical trials, and not just the kind of the big well-designed ones where there's a lot of stats input. I wonder if people have any thoughts.
Simon, did you have the follow-up on that, or as in, would you like to come in and then we can put it to the panel if there's anything you wanted to add?
Well, thanks. I have a comment. I'm not sure if it's a follow-up or if it's an answer. It's somewhere in between. But actually, I'm not sure that the Biedermann's sort of framework particularly allows you to leave patients out And one of the problems that I have with intention to treat is all it does is say that every patient has to be in the analysis, but it doesn't tell you how, and it doesn't tell you what to do with data from patients who did funny things here and there, whatever they may be. It just says leave the patient in. So ITT, it's a principle that I certainly stand by, but it doesn't actually give an answer, whereas the S-demands framework, I think, tries to get you to think about, assuming you are going to leave all these patients in who have done all these naughty and funny things, how are you going to do it? So I'm not sure that necessarily it is pushing the door open a little bit, but others may disagree.
Okay, does anyone want to go first on the panel to address those?
Yeah, I can start. So I share a little bit this this concern that we kind of weaken ITT, and so I think we need to be careful about that. So that's one thing. On the other hand, I've been part in trials where you have recruited 1,000 follicular, or the population of interest is follicular lymphoma patients. You recruit 1,000 patients, and then when you look at the data, you find 40 have diffuse large B-cell lymphoma, a very different disease, shares some kind of treatment strategies. I think it's a fair question to ask. We are interested in the follicular lymphoma population. Should we include those 40? The way we've run trials at Roche always is there is no discussion about that. These patients are included. But that's a baseline diagnosis. It's a different disease. This is when sites rush to randomize patients before they have proper histology done, etc. So that's a fair question to ask. Should we really just include them by default? So that's one thing. The other thing is, as I hopefully made clear a little bit with this treatment switching example, primary interest might not always be just what we call ITT or treatment policy. There might be really questions where maybe we should deviate from that. And then to Simon's point, I just pulled what ICHE9 actually says. And it seems we forgot that, because I read here, this is a quote from ICHE9. The intention to treat principle implies that the primary analysis should include all randomized subjects. That's typically what we say is ITT. Continue the quote. Compliance with this principle would necessitate complete follow-up of all randomized subjects for study outcomes. This is typically what we ignore with ITT, because if we did that, we would not need ICHE9 addendum. The problem is, until this hits reality, in clinical trials, you will not have complete follow-up of all randomized subjects for study outcomes. And the original ICHE9 guideline is completely quiet on this. And the addendum is basically fixing that second part.
Thank you. Does anyone else have any comments, panel?
I think it's just to say that, well, I guess my bias in terms of how I would see this will come largely from a large portion of what I know on S-demands has come from listening to Kaspar talk over the years. And so maybe we'll end up with a similar viewpoint on that one. But I guess either way, in terms of the way I see it, and this is maybe a little naive to the reality, but in both cases, you're relying on the principled statistical side of the approach. And so ultimately, whichever, whether you're using S-demands or following the ITT principle, actually it's still kind of, the reliance is still in the same place in terms of making sure that your approach is principled. And so ultimately, does it really change from what came before on that basis? I would argue maybe not. It's just a better way for us to then be able to communicate how we're being principled, is what comes through the addendum. I don't know if maybe that's not a fair comment, but I'm sure Kaspar can correct me if I'm wrong on that one.
No, I think it's true. It doesn't change principles per se. It just fills gaps. I mean, I have a huge respect for ICHE9. If something like this is in effect unchanged for more than 20 years, this is a huge, huge achievement. But now we've come to realize that, yeah, maybe we should talk more about what happens if you don't have complete follow-up of all randomized subjects, and try to give some guidance. And I mean, it all came out of the discussion for longitudinal data. And now we just now detect and find what holes we had in this whole theory for longitudinal data in the past. And there is huge efforts are going on to kind of implement methods and make sure we are clear what we are estimating. We're just not taking MMRM off the shelf, apply it, and all the missings, the model takes care of them. Yeah, it does, but it does so under quite stringent assumptions. And maybe we should also have a discussion about all these assumptions. And maybe for some patients they apply, for others they don't. And for that I see huge benefit. And I mean, these developments are not immediately caused by the addendum, but they are now implied because people start to think much more carefully about what they have done in the past. And so this has all been kicked off by the addendum, and maybe to an extent that not even those who have written the addendum could foresee.
Great, thank you very much. I think Mona has also shared some experience. I don't know, Mona, if you sort of have you sort of experienced some sort of similar sort of issues to what have been being talked about?
Absolutely, I think in terms of communication and the need of a statistician, and and it's not just the methods that we need to be involved right at the beginning and understanding all the ins and outs, which I think some clinicians fail to see, and they just draw on our experience right at the end, basically, which I think sometimes can be too late. So this is something that I very, very recent that I've encountered, where it's been around for so many clinicians, basically what they were trying to do, and they missed key elements to think about. So it's not just how you analyze it. I'm not there just to confirm. I was approached just to confirm the sample size, and it's and there were so many holes basically in the thinking of it that I had to go back to them, and they realized, okay, oops, we missed this and that. So it's important, that dialogue and bringing us in terms of the thinking about the design rather than just the mechanism of doing statistics. Thank you very much. I'll stop there.
Thank you very much, Mona. So we have a question for Stefanie. I'll bring Richard in to ask that. I'm not sure if this is because obviously I've worked with Stefanie on this topic, so I was also thinking about it myself, and let Stefanie answer, of course, as well. But I wonder if this is a special case of not missing at random from what we're talking about. So, but I'm not sure. So Richard, do you want to elaborate on that, and then we can ask, and Stefanie can respond.
Yeah, sorry, I'm not trying to monopolise the questions. I'll ask quickly. I was thinking about your example trial with the 4 arms, with the Alzheimer's one, and then you reanalyze it based on the observed amount of missing data within each of the arms. And I wonder how you deal with a scenario where the amount of missing data, as Robin just said, is kind of missing not at random, or linked to the effectiveness of the treatment, so that you get less outcome data in the efficacious arms, or more outcome data in the case of cognition in Alzheimer's. Like, how do you take the effect of treatment on the proportion of missing data into account when you reanalyze the trial to establish your optimality criteria?
So yeah, let Stefanie answer. I'm going to say a short answer will probably be with a lot of difficulty, I suppose. But I'll let Stefanie respond.
Yeah. So the analysis we did was mostly for getting some model for the dropout in order to do the design, but still that was under the assumption of missing at random, so that we would have missing at random given we know what treatment the patient got. And what happened was that it was the placebo where we had the most dropout, and I I do think it has something to do with the effectiveness of the treatment, that essentially those who had the placebo they had no benefit from the placebo, obviously, and then their Alzheimer's progressed more, and they were more likely to not be able to attend the next clinic.
I think we have looked into sort of not missing at random, not in that level of complexity in a simpler setting. It's very, very challenging, obviously, not missing at random. And then I think, obviously, we sort of, like you do in design, you need to conjecture a sort of model, and then you can look at maybe how good that model is. So you could, for example, I imagine the framework would allow you to put in outcome into the missing probability or propensity to be missing, but you would be very sensitive to the actual model specification, as you could be in other scenarios as well. So at least from my perspective of missing data, which is the area I'm in primarily, for those kind of problems, you often do like sensitivity analysis. But yes, I mean, I don't think there's an easy answer to that. But I'm not sure if anyone's aware of in the clinical trials literature anything more that's done on that. But it seems to me to be this informative missingness problem is a real challenge in all areas, really. Okay, I don't know if anyone. I don't think I've missed any questions. Feel free to jump in or tell me if I have. It was really great discussions to have, and lots of great feedback and conversations. So thank you everyone for attending. We have come to sort of the end of our session now. The session is recorded, and I think it should be made available sort of by end of January. I think there's a policy, if people wanted their parts removed, you could email the RSS team to ask about that, but otherwise it should all be posted without redactions otherwise. And yeah, I mean, it's really thank you very much to everyone, to the speakers, and for presenting. It was some really great talks, and thank you everyone for attending and asking questions and interacting.